Hey there, science fans. Today we're diving into the amazing world of DNA and RNA. These two molecules are the fundamental building blocks of life as we know it. They hold the instructions for everything that makes you, you. From the color of your eyes to your height, it's all thanks to DNA and RNA. Think of DNA as the blueprint of life. It contains all the genetic information needed to build and maintain an organism. RNA, on the other hand, is like the messenger and the builder. It reads the DNA blueprint and helps create the proteins that carry out various functions in our bodies. Without DNA and RNA, life as we know it wouldn't exist. Pretty cool, right? Let's take a closer look at these incredible molecules. DNA, short for deoxyribonucleic acid, has a unique shape. It looks like a twisted ladder, which scientists call a double helix. Each rung of this ladder is made up of pairs of chemical building blocks called nucleotides. There are 4 different nucleotides: adenine, A, thymine, T, guanine, G, and cytosine, C. These nucleotides always pair up in a specific way, A with T and G with C. We call this complementary base pairing. This unique structure helps DNA store vast amounts of genetic information in a compact and elegant way. It's like a super efficient library where each book contains instructions for building and running a living organism. One of the most important things DNA does is make copies of itself. This process is called DNA replication, and it's essential for life. Every time a cell divides, it needs a complete copy of DNA to pass on to the new cell. During replication, the DNA molecule unwinds and separates like a zipper unzipping. Each strand then serves as a template for building a new complementary strand, following those special base pairing rules. The result? 2 identical DNA molecules, ensuring that genetic information is accurately passed down from one generation to the next. It's like photocopying the blueprint of life, so each new cell has all the instructions it needs. Pretty neat, huh? Now, let's talk about RNA, or ribonucleic acid. RNA is similar to DNA, but with a few key differences. First, it usually exists as a single strand, not a double helix. Second, instead of thymine, T, RNA uses a nucleotide called uracil, U. RNA plays a crucial role in protein synthesis, which is the process of making proteins. You see, DNA lives inside the cell's nucleus, but proteins are made outside the nucleus, in the cytoplasm. That's where RNA comes in. It acts as the intermediary, carrying genetic instructions from DNA in the nucleus to the protein-making machinery in the cytoplasm. Section 5. From DNA to protein. Transcription and translation. This is a fundamental process that occurs in every living cell, ensuring that the genetic information encoded in DNA is accurately converted into functional proteins. The process of getting from DNA to protein involves 2 main steps: transcription and translation. These steps are crucial for the expression of genes and the production of proteins that perform various functions in the body. Transcription is like making a copy of a recipe from a cookbook. It's the first step where the information in a gene's DNA is transferred to a messenger RNA, mRNA molecule. During transcription, a special enzyme called RNA polymerase reads the DNA sequence and creates a complementary RNA molecule called messenger RNA or mRNA. This process ensures that the genetic code is accurately copied. This mRNA molecule carries the genetic code from the DNA to the ribosomes, which are the protein factories of the cell. The mRNA acts as a messenger, conveying the genetic instructions from the nucleus to the cytoplasm. Next comes translation, where things get really interesting. This is the step where the genetic code carried by the mRNA is decoded to produce a specific protein. During translation, the mRNA molecule is read 3 nucleotides at a time. These 3 nucleotide sequences are known as codons. Each 3 nucleotide sequence, called a codon, codes for a specific amino acid, which are the building blocks of proteins. This coding ensures that the correct amino acids are added in the proper sequence. The ribosome reads the mRNA codons and links the corresponding amino acids together in a chain, following the instructions encoded in the mRNA. This process is essential for the synthesis of proteins. This chain of amino acids then folds into a specific three-dimensional shape, forming a functional protein. The final shape of the protein determines its function in the cell, allowing it to perform its specific role effectively. Section 6. The many roles of RNA. RNA is a versatile molecule with many different roles in the cell. It is not just a simple messenger, but a key player in various cellular processes. We've already met messenger RNA, or mRNA, which carries genetic information from DNA to the ribosomes. This process is crucial for the synthesis of proteins. But there are other types of RNA too. Each type has a unique function and contributes to the cell's overall operation. For example, there's ribosomal RNA, or rRNA, which is a major component of ribosomes themselves. Ribosomes are the cellular machines that assemble proteins. Then there's transfer RNA, or tRNA, which helps to bring the correct amino acids to the ribosome during translation. This ensures that proteins are built accurately. Each type of RNA plays a critical role in ensuring that the instructions encoded in our DNA are accurately translated into functional proteins. Without these RNA molecules, the process of protein synthesis would be impossible. Think of it like a well-coordinated construction crew. Each member has a specific job to do, and they must work together seamlessly. mRNA brings the blueprints, rRNA builds the factory, and tRNA brings the materials. This collaboration is essential for the cell's function. Together, they build the amazing proteins that make life possible. These proteins are involved in virtually every cellular function, from metabolism to cell division. Section 7. RNA and DNA in action. Modern medicine. Scientists are constantly making incredible discoveries about DNA and RNA, the fundamental building blocks of life. These discoveries are leading to groundbreaking breakthroughs in the field of medicine, transforming how we understand and treat diseases. For instance, gene editing technologies like CRISPR-Cas9 allow scientists to precisely target and modify DNA sequences, offering unprecedented control over genetic material. This groundbreaking technology holds immense potential for treating genetic diseases, developing new therapies, and even preventing inherited conditions. RNA is also playing a starring role in modern medicine, with its versatile functions being harnessed in innovative ways. mRNA vaccines, like those developed for COVID-19, have revolutionized vaccination strategies, offering a faster and more adaptable approach. These vaccines deliver mRNA instructions to our cells, enabling them to produce a harmless piece of a virus, which then triggers an immune response, effectively teaching our immune system to recognize and combat the real virus. This approach has the potential to be safer and more effective than traditional vaccines, reducing the risk of side effects. It could be used to target a wide range of diseases in the future, from infectious diseases to cancer. The advancements in RNA and DNA technologies are paving the way for a new era in medicine, promising more personalized and effective treatments for patients worldwide. Section 8. The future of DNA and RNA research. The future of DNA and RNA research is incredibly exciting. Scientists are constantly developing new technologies and approaches to understand these molecules in even greater detail. For example, new sequencing technologies are allowing us to read DNA and RNA sequences faster and more affordably than ever before. This is leading to a deeper understanding of the genetic basis of disease, personalized medicine, and even the potential to bring back extinct species. The more we learn about DNA and RNA, the more we realize how much more there is to discover. Who knows what amazing breakthroughs the future holds? Section 9. DNA and RNA, the building blocks of life. So there you have it, an inside look at the incredible world of DNA and RNA. These two molecules are essential for all forms of life, carrying the genetic instructions that make us who we are. From the double helix structure of DNA to the intricate dance of transcription and translation, the processes involving DNA and RNA are nothing short of remarkable. Understanding DNA and RNA is crucial for advancements in medicine, agriculture, and many other fields. The more we learn about these molecules, the better equipped we are to address some of the world's most pressing challenges, such as disease, hunger, and environmental degradation. Section 10. Conclusion. Our microscopic marvels. DNA and RNA might be microscopic molecules, but their impact on life is anything but small. They are the architects of our very existence, the keepers of our genetic heritage, and the driving force behind the incredible diversity of life on Earth. So the next time you look in the mirror, remember the amazing molecules working behind the scenes to make you uniquely you. DNA and RNA are true marvels of nature, and their story is just beginning to unfold. Keep asking questions, keep exploring, and never stop being curious about the world around you.